who was actually my lab mate at the time. Wasn't quite faculty lit, but now he's faculty. And then after that, I said we also had in common, we spent time in Panama while we were in Princeton, so we got to do that. So just, the same journey, just like, it was about a dozen years different maybe. That's why my hair is so gray. And then after Alexa finished her PhD, she went and did a postdoc. And so she actually went back to New Jersey, but to Rutgers this time. I refused at that point to leave Santa Barbara because I liked it so much. So she did a postdoc. I assume that was with Malin.
That's right.
With Malin Pinsky, who followed you, I guess, now to UC Santa Cruz, and is here now as well. And so a little bit about Alexa's research interests. So she does a lot of really amazing work, mostly computational, working with big data sets, trying to understand particularly the effects of climate change on species distributions, how it's shifting species ranges, really digging into like what's happening at the warm end of the range, the cold end of the range. Are species tracking climate in their distributional shifts? Are they responding to short-term things like marine heat waves or not? So some really cool work there, mostly with fish. And then other things that and I think that's mainly what she'll tell us about today, but she's also really interested in like open data science, preserving big data sets, access to data, and so that's kind of another big push of things that she's interested in. So with that, I'm really psyched to hear your talk, and take it away. So Alexa's going to tell us about beyond simple models of marine species on the move.
Thank you so much for the lovely and very detailed introduction. You covered some things I won't get to talk about, so you can ask me about those later. And if anyone has trouble hearing me at some point, please just raise your hand and remind me to speak up. I'm so excited to be here visiting a neighbor, and I really thank you for the invitation. This is something I've been really looking forward to. So Scott did a great job of summarizing my research. I work on the question, why are species found where they are and not elsewhere? So all of my science fundamentally is about this question of, when we look around us, why do we see the stuff that we do see, and why don't we see other stuff? And then the related question is, what makes species move? Broadly, this is the field of biogeography. This has been around since Charles Darwin and Alfred Russel Wallace. There are some really fun books in my office from the 1800s that talk about this exact question, if you're ever interested in visiting me or asking me about them. This question, though, takes on new urgency now because at the moment, species are on the move around the world. And they're not moving randomly. On average, species are moving upwards in elevation on land. They're moving deeper into the oceans. And globally, they're moving towards the poles. So this has brought the research question of many centuries about understanding why species live where they are, not elsewhere, into kind of a very specific focus in the Anthropocene. This has major consequences for biodiversity and conservation. In this part of the oceans, we have not had as directional of a shift in the distribution of our kelp. But in other places, like the Australian continent, kelp has receded really, really linearly down both coastlines as it's gotten warmer. And of course, that takes with it all of the services that a kelp forest provides to nature and people. These shifts also really challenge the way we manage natural resources. I have a little bit of a background working in groundfish management, which I won't have time to get into, although happy to answer questions about it. This was a species that was a very hot topic when I worked in groundfish management. It's a canary rockfish. They were overfished in the 90s and declared rebuilt in 2015. But when they rebuilt, they were in a totally different part of the West Coast than where they had been overfished from. So it makes it really difficult to manage marine populations, particularly the ones we harvest, if we literally don't know where they are or why. And also, species that are moving around challenge fundamental parts of human culture, society, and history. Particularly indigenous communities have often a really close dependence on species being in a place at a time. These are indigenous families in Finland that harvest walruses at a specific time of year on sea ice where the walruses are no longer really found. So species in the move affect people in many different ways and ecosystems around the globe. So I also work a lot on the consequences of these shifts for nature and people. Don't have that much time to talk about the people side today, but ask me about it if you're curious. So this is a brief outline of today's talk. We're going to start with an introduction to species in the move. So if you've never heard of this phenomenon, don't worry. It'll make a lot of sense in a few minutes. I'll talk about the work I've done kind of at the scale of whole marine fish communities, and then we'll zoom in to think about species range edges, and then even more to think about the mechanisms underlying these changes at different scales and better ways to model and measure them. So I usually introduce the idea of species in the move by talking about changes in space and time, which will start with space, because that's usually what we think about when we talk about things shifting. So we know in the oceans that species are shifting their distributions quite rapidly. One of the seminal studies on this was done by my postdoc advisor, who, as Scott said, is now at UCSC as well. This is a study where they took data on hundreds of taxa from North American surveys from Canada and the United States of the continental shelves and looked at how rapidly species are changing their distributions in terms of degrees latitude. And that's on the y-axis. And then they asked for each of those taxa, where would we have expected them to shift if they were staying at a constant temperature? So if they're shifting north, how far north would they need to go to stay at the same temperature in a warming world? That's the red line. That's a one-to-one line on this plot. And what you see here is across hundreds of taxa and different oceans, there's a really strong correlation between how far we expect something to shift in a warming ocean and how far it actually has been moving. This is all historical data. This has already happened. Studies like this and hundreds, thousands of others have been synthesized into these really big papers about species in the move. And this is a particularly fun one, which I'll take a second to tell you about. I'm going to hop up on the real stage. So this is a study that took every published paper about any species shifting its range anywhere at the time they wrote it in 2020. They separated them by taxonomic group. Blue is ectotherms. And the thing I want to draw your attention to is the bars on the right. So over here, you can see that where it says T is terrestrial, where it says M is marine. And they're ranked by how fast they're shifting north. So the faster they're shifting north, the further to the right they are. We should congratulate ourselves that the oceans are winning the range shift Olympics by a lot, right? Most of you, I think, no matter where you are in this program or in your careers, recognize many of these names. These are all marine species in the bottom of this plot. And this paper with great fanfare updated the rates of shift for the globe, saying that on land, things are shifting on average 1 kilometer per year. In the oceans, it's 6. That's actually very fast. That's 60 kilometers per decade. That's more than half a degree of latitude. The other thing to notice, though, is how wide these distributions are. So those kind of sort of oblong shapes that each taxon has is the distribution of all of the data this study synthesized, and they go from things that are shifting ridiculously fast towards the poles to things that are actually running towards the equator. So this represents an enormous amount of variation in addition to a really large trend. Species are also in the move in time, which is a slightly more complicated thing to conceptualize. And there are a couple ways that this manifests. One of them is that over time, communities in the oceans are changing in a directional way. This is a great image of that from another fish survey in the Barents Sea. The color of the dots represents how relatively cold affiliated the fish community at each site is. They call the cold ones Arctic-like, those are blue, or how warm affiliated they are. Those are the boreal-like ones in red. And you can see here that in 8 years, the whole ocean in this ocean region in this survey had an enormous amount of turnover to be very warm. And the cold community got really concentrated towards the very, very northern part of what is a super high latitude survey. So this process is called tropicalization, a very fun and intuitive word, I think. So that's one temporal form of change that we've seen. But another one is things are literally shifting their life history and timing to be earlier or at a different time of year. These are data from salmon runs, looking over decades to see when the peak salmon date was. And on average, many of these species have shifted that date, I don't know, up to a week or so, or a little bit more earlier in the year. This is mostly described for terrestrial species because we see it a lot in plants, in insects, and other things that kind of have a life cycle and life history that's very annual and very tied to environmental cues. But in anything with that kind of life history, we see those cues shifting earlier and earlier, which is called a phenological shift. So these are all species in the move, and they're really correlated with climate change, which sort of begs the question of, are we done? can we say that we have a really strong ability to predict and infer future changes about climate effects in marine systems? Unfortunately, we actually have very low predictive skill from a modeling standpoint from these kinds of inferences, which has been a puzzle in my field in recent years. The correlations are all really strong, but it's really difficult to actually make predictions for the future that are correct. These are, I could have put many papers up here. These are some favorites. If you choose one to read, I like Paintings Predict the Distribution of Species, which is a paper that uses paintings as inputs to species distribution models and finds that they outperform environmental data, which is bad. So I think it's worthwhile to dig into what's actually happening at different scales and understand these phenomena in marine systems. So we're going to start by doing that by saying, well, if species are on the move in the way we expect them to be in the oceans, what would that mean for communities? At short time scales, what we expect to see is like that image of the Barents Sea, where if we have an extreme climatic event like a heat wave, we should see turnover towards warm affiliated species, which would, like the heat wave, they'd do well, and the cold affiliated species would do poorly. At longer time scales, we would probably continue to expect to see that tropicalization process. We'd also expected related but not the same phenomenon called biotic homogenization. And I'll explain each of these in more detail later, but biotic homogenization means that communities across space, so let's say the ocean community in Monterey Bay versus the ocean community down south in San Diego, start to look more like one another because the same species are kind of spreading out everywhere. So we've tested for both of these, and I'll start with the short time horizon one. So after a marine heat wave, I think what many people imagine happens to an ecosystem is that cold affiliated things do really badly. Maybe they die, maybe they leave, maybe they fail to reproduce or fail to grow. It can actually be any of those because what we're measuring is just biomass, but they are not happy. And the warm ones are fine. But that's not the only possible outcome. You could also see, okay, the cold ones do badly, but the warm ones actually do really well. They're happier during the heat wave. That could happen, and the cold community is fine, which would lead to a net increase in biomass. And of course, you could see neither, and the community just stays the same. These are actually, as you can probably see, testable hypotheses about the community composition and the biomass that we see in the oceans before and after extreme events. So I tested these using a giant data set of bottom trawl surveys. So this is when usually federal agencies go out and go fishing with bottom trawl fishing gear, record everything that they catch, and then a bunch of unpaid grad students and postdocs spend like 5 years harmonizing those data so we can do stuff like this. This is 18 surveys, 80,000 net hauls, 22 million discrete rows in a data frame that say, you know, a fish was here, yes or no, and how much of the fish was here, and 248 bottom heat waves. So it's from the subtropics to the Arctic. It is a very large data set. And the project that generated this is called Fish Globe, which I'm happy to answer questions about too. The data are public. You can use them. So to answer this question about impacts, we calculated something called the log ratio of biomass, which means how much biomass do you have this year versus last year. If it's a positive number, you got more. If it's a negative number, you got less. And we hypothesized that as marine heat waves got more intense, that's what's on the x-axis here, you would get a loss of biomass. And when there was no marine heat wave or smaller marine heat wave, you would see kind of as likely to gain or lose biomass. Instead, we saw no signal whatsoever. So as heat waves get more intense, there was no evidence that biomass on average went down. And to convince you that this is a real result, we have some of your favorite heat waves to talk through. Many of you are familiar with the warm blob. The West Coast survey doesn't light up on this one as much as the Gulf of Alaska 2017 survey, which is one of the most intense heat waves ever in this data set. And you do see here that biomass was lost in that survey, so each dot on this is a whole survey's fish change from one year to the next. They did lose biomass, and that's right. There were a lot of fishery collapses in that part of the oceans between 2015 and 2017, which is that interval. But if you look for 2012 up top, when there was a huge heat wave in the Northwest Atlantic, you see that the Northeast US survey and the Gulf of St. Lawrence survey, that's in Canada, both had huge spikes in biomass. And that's also correct. Those were the best years ever for fisheries in those regions at that time. Overall, if you compare, was there a heat wave, yes or no, the average biomass change is 0 from one year to the next, so you're as likely to gain fish as you are to lose them from one year to the next, regardless of whether or not there was a heat wave. For the second question, do we see tropicalization, turnover towards warm affiliated fish? We calculated that same log ratio, do you have more fish this year than last year, but now it's for every species, so many dots per survey. I'm only going to show you things after a heat wave. And the way we calculated, is it a warm affiliated fish or a cold affiliated fish, is we asked, what is this fish's favorite temperature relative to the average favorite temperature in this survey? If the fish liked warmer water than the survey on average, that means it has a positive thermal bias, so that's towards the right of the x-axis. So we expected these points to have a positive slope. So you have warm affiliated fish doing well in the heat wave, cold affiliated fish doing poorly. Instead, we had no pattern whatsoever. The dots show you the intensity of the heat wave. So these are all heat wave years, but different intensity. And also, if you look across, just was there a heat wave, yes or no, and average all these species to say, well, okay, maybe there's no species level pattern, but on average, does the fish community look more warm affiliated during a heat wave? The answer is also no. I used this color ramp on the left as a joke, and none of my co-authors told me to take it out. So it is in the paper. We call it the Barbie plot. So I'll happily answer questions you have about these results later, but I will just say they're extremely robust. After 4 rounds of review with 4 reviewers, we tried every possible way we could think of to find a relationship between the amount of fish in groundfish surveys and heat waves, and it's extremely robust that there wasn't one. You can ask me more anytime. So that's our short-term answer to, do we see the fingerprint of climate change we expected on marine fish? Now let's look for the long-term one. I told you about this concept called biotic homogenization, which basically means that when you have a broadly distributed stressor to ecosystems like climate change, if you start out with something on the left where it's kind of hard to see, there are some fish people here who will recognize that these silhouettes are different groundfish. But these are all communities that don't have any of the same species. They're quite dissimilar from each other, even though they're next to each other. And the theory of biotic homogenization says, in the context of something like climate change, you would expect them to get really similar, because you sort of have the same winners and losers everywhere. So you don't necessarily have a huge loss or gain of biodiversity overall, but what you do get at the site level is much less uniqueness. And obviously, this is not something that we would necessarily want. Having unique sites gives us a lot of ecological resilience and also just as part of biodiversity. So we tested, using the same data set, whether we saw this trend over time towards less dissimilarity, which is the insanely confusing term ecologists chose to describe this process. So the dissimilarity metric is here on the y-axis. It doesn't matter what the number is, but each line is a trend in one of those surveys. So if things were getting more similar to each other over time, that line would have a negative slope. There are some of those are yellow, but about a third of the surveys had a negative slope, about a third had a positive slope, so they're actually getting more differentiated over space. Those are pink, and then the ones that have no change are blue. This study actually, they got even more data than I did. This includes 20% of the world's continental shelves. So there are more than 18 surveys on here by a lot. I'll show you the West Coast in case you're interested. Our local survey is right there. It's a small dot because it hasn't been running for quite as long, and we have a blue, so there's no net trend towards either more or less similarity over space in several decades of environmental change. So at the community scale, what we see here is that marine heat waves don't have the general effect that we expected at short time scales on marine fishes. I didn't show you plots for this because we're friends, but my postdoc has been fitting this really fun statistical tool called empirical dynamic modeling, which basically is designed to detect nonlinear causal relationships in high dimensional systems. to the longest survey time series of all of the many that I've showed you. It happens to be New England's NOAA bottom trawl survey, to see if we can detect sort of a much more complex relationship between marine heat waves and fishes. Also, the answer is no, basically, for almost all species. Maybe one or two species with very short lifespans that kind of have a cyclic behavior in response to their marine heat waves, but that's of dozens and dozens of species that we've tested. And also, this other long-term hypothesis that we should see biotic homogenization at large scales also isn't really supported in demersal fish. Usually at this point in the talk, people seem very discouraged by my line of work, but I actually think that this shows us something really positive, which is that these species are incredibly resilient. Most of our stories about climate change impacts on marine life don't come from places like this. They come from coral reefs and a lot of places that do have much worse and much more consistent responses to the warming and heat waves that we've had so far. That's not to say that there haven't been problematic effects off of our coast. Many of us know of many of those. But actually, when we look at demersal fishes, and these support most of our global major fisheries and a huge amount of biodiversity, they're actually really resilient to short-term heat waves, to long-term change, at least as far as we have collected data. So this kind of, to me, begs the question of, how well did we think these things were going to be tracking temperature, given that we don't see the temperature fingerprint that we thought we would? So this is the part where I get to talk to you about species ranges, which is my favorite thing to talk about. The reason that we all thought that species in the oceans should have this really clear climate change fingerprint is because of a connection between physiological lab experiments on fish and the global geographical distribution of fish. If you take fish, put them in a lab, you cook them, you freeze them, you can tell I have no idea how lab experiments work, and you get their thermal maxima and minima, and then you actually go out and look on a map and say, okay, well, where are the extreme limits of the species distribution? The temperatures at those places are almost exactly the same as what you find in the lab. And that's what this figure is telling you. It's telling you that both at the warm and cold boundary of marine ectotherms, they live right up against their thermal limits. And that strongly suggests that the moment temperature changes, they should respond, because it seems that their biogeography is really strongly connected to their physiological thermal tolerances. So that was this paper and some related papers found that really close connection, and it has inspired sort of an assumption in many models about the oceans that fish and marine invertebrates and all marine ectotherms are should perfectly track temperature. So I tested this using, this was before the Fish Globe project, so I only used 3 of these bottom trawl surveys, to see, well, do we actually see those warm and cold boundaries shifting year by year as temperature changes? And when I did this, I ended up with 165 species range edges, which was a lot more than anyone had previously used to look for the same pattern across many species. These 3 regions are particularly fun to look for responses to environmental change because they have had really different warming histories. The Northeast US, I talk about a lot because it has just had monotonic, really intense warming. So it's warmed over 1 degree Celsius in less than 50 years. And that mostly has happened through summer warming, it's the red line, and average warming, which is the orange line. Winter temperatures there have not really warmed. Our region, the West Coast, has not gotten warmer in the time series that we have for the continental shelf bottom. But we have enormous environmental variability due to the PDO, El Nino, and other phenomena. And the Eastern Bering Sea has both. It has warmed there, but non-linearly, through a combination of these sort of warm and cold stanzas that recur. So the question is, are species actually following those wiggly lines in every one of these regions? And I tested this by saying, okay, well, let's look at how far the species shifted. So I keep showing you figures where we say, okay, this is how far the thing moved per year, because that's sort of how we think about species distributions. That's on the x-axis. But now what's on the y-axis is how well it maintained a temperature at that range edge. So if it's perfectly tracking temperature, it should be at the horizontal 0 line all the time. Because whatever happened to temperature in that region, the species range edge perfectly followed it, up and down the coast or wherever that took it. We did this for both summer and winter temperature because I thought it was more likely that a species was limited by extreme temperatures and averages, but you don't actually know which extreme. So we tested both. Here are the data for those 165 range edges. And the main thing that jumps out is that almost all of them actually overlap with 0. If you want to know how to put a confidence bound on a range edge, you can ask me, but I'm not going to go that deep into the stats now. So it looks like they're actually tracking their thermal niches really well because they are staying at about the same temperature over many decades as temperature kind of wiggles up and down. But the species that aren't tracking temperature very well are in the place that has had the longest and most consistent warming, which is the Northeast US. And this was really troubling because it suggests that maybe the other species, it's not so much that they're actively tracking temperatures, that we don't have enough data and enough warming to disprove the null hypothesis that they're tracking temperature. And in the place where we do have that much data, which is New England, that relationship kind of breaks down. So it's also possible that species are tracking, so I should say this was surprising to us. We expected to find much, much closer temperature tracking, especially in New England, a place where species are moving up the coast really quickly. It's sort of a poster child for marine species in the move and fisheries management of species in the move. And so then we thought, well, maybe we've kind of misrepresented the way range shifts are happening. Maybe it's not so much that we need to look from 1 year to the next over 50 years. Maybe we need to think about how species actually experience environmental change and what they do next. And it's possible that range edges are moving less by just sort of marching along and more through kind of sequential responses to extreme events, like heat waves or long-distance dispersal events. So the cartoon on the top is sort of what most people think, that species ranges sort of march along as the environment changes. But maybe what actually happens is that these recurrent extreme events either cause species at the warmest edge of their range to kind of die back and then maybe rebound a little bit and die back and rebound. And at the furthest end, usually the poleward edge, maybe they have periodically opportunities to go really far, and then they might or might not survive. Maybe they bounce back, and then they do that repeatedly. This is almost impossible to test for in data because species ranges are huge, and you don't really have a systematic survey that would capture something, for example, moving hundreds of kilometers and then vanishing and moving hundreds of kilometers and vanishing. So to answer this question, we instead turn to, first of all, marine invertebrates, which are really dispersal limited in their biogeography, unlike fish, which annoyingly can swim. And we also turn to historical records to try to build a data set that would let us answer this question of how much are range shifts actually happening mostly through recurrent extreme events. So this is a fun project for me to share because this was done by my grad students. We looked through, we did an enormous literature search and also read all of the historical reports dating back to the 1950s from Cal Coffey, here on the left. And records going back to 1900 of any time someone wrote down that they saw a weird marine invertebrate either turning up in California that isn't normally here, or a weird Californian marine invertebrate turning up really far north. So we had fun records like this one. On the morning of August 6, 1934, much excitement prevailed in the San Pedro wholesale fish markets due to the arrival at the quayside of a local ringnet with 3.5 tons of a species of squid entirely unknown to the San Pedro fishermen. This is jumbo squid, extending its range a very long distance in 1934, and it turns out jumbo squid has actually done that repeatedly in the past century. This is work led by my PhD student, Jordana Savigny, and she's submitting this paper in the next month or so. So I'll show you some of her results, which I think are really fun. The first thing we tested is, okay, well, how often would we expect? So our main question, because we're trying to understand, are range shifts driven by recurrent long-distance range expansions? In this case, that means El Nino, right? When we think about weird conditions driving stuff really far north, we think about El Nino. So what we wanted to know was, well, how many of those range extensions are happening during El Nino? In our data set, so 125 years since 1900, about a quarter of the years were El Nino years. So our null hypothesis is that about a quarter of those range extensions should happen during El Nino. What we actually see is that it's way more. Each of those dots is a species that extends repeatedly in the data set, so they're kind of recurrent extenders that are appearing, dying back, appearing, dying back. And almost all of them occur way more frequently in El Nino years than you'd expect by chance. In case you're wondering who these are, and you're a marine invertebrate person, the ones that have 100% of their range extensions during El Nino are, there's a krill species, the Velella by the wind sailors, pelagic red crabs, and mole crabs. And then on the other end, the one that has 0% of its range extensions during El Nino actually is a gastropod that has a really well-documented range extension that's sort of an ecologically mediated invasion in California that has nothing to do with El Nino. That's a Mexicanthina snail. In addition to being more common during El Nino than expected by chance, our data set of range extensions clusters in big El Nino years. So the bubbles are the range extensions. The time series plot shows El Nino regimes in red and La Nina in blue. And you can also see how big these circles are. The circles show how many weird range extensions there were in this recent period that are associated with the blob. Almost a quarter of all of our data come from just the blob years. We also found that when the El Nino is stronger, which is on the x-axis, things move further and more stuff moves. So the frequency of range extensions and the magnitude of range extensions is also really related to El Nino. So our kind of take-home messages here are that range edges are highly correlated with temperature, but not in a way where we can probably use that relationship to predict where they're going. Whether the long distance extensions that are driven by El Nino are more predictable, I think is kind of unknown at this point. We know that they're really related from the results I just showed you, but I don't know if I could tell you, okay, we have an El Nino this year, are we gonna see pelagic red crabs, yes or no? That's a hard question to answer. So to answer that question, we need new models and new methods. And I know we are getting towards when you normally start your Q&A, so I'll try to do this part kind of quickly so that we have plenty of time to chat, because I'm sure people have to go. So I'll just briefly say that, okay, I lied. It's not going to be super brief, just to prepare you, but I'll try to be quicker than I would have been. So when I see species ranges, and when I ask people what is a species distribution, most people are thinking about the same thing, right? When I say those words to you, you're picturing something, and it's about what I'm picturing. Most of us are picturing sort of a blob on a map, that's the range, and then the edges are the boundaries of the blob. And that concept is widely shared in ecology and marine science. If I ask you, what is this species range, based on the data on this map, which are the actual data from observations of a canary rockfish, I would get as many answers as there are people here. And I'm gonna argue and try to convince you that this is really a problem. And here's why it's a problem. Everyone, and this is a critique of my own work first, just makes up metrics for this stuff that makes sense to them, right? They're like, I think it's the 95th percentile of latitude, or like, I'm gonna fit a convex hole to the data and use a biomass weighted thing, or something like that. Please come back. I can unplug this one, but it'll like undo the thing, I think. Yeah, looks better. Yay! Just your presence. Thank you. So we all make up, essentially, metrics as researchers for what we think makes sense for how to describe a species range, which seems innocuous. I have done it. But the problem is that then kind of the way ecology works is that we aggregate those kinds of studies and use them to inform our global understanding of climate change effects. So that plot I showed you on the right earlier is exactly doing that. It's aggregating all these studies. And then we have this kind of recurrent problem that has been really widely described in the past few years, which is that we can't actually predict climate change responses of any species. And there is as much variation related to the researchers and the study as there is related to environmental drivers and climate change. So we can't actually really describe global species on the move very accurately because we don't really know what being a species in the move means from a quantitative standpoint. So I'm going to try to briefly convince you of two things: that our methods affect our metrics for species in the move, and that we may need more data to do a better job of this than we often have. So I'll skip this one and just show you, because that's a bird and none of you care. So this is, so here's a fish, and I took data on the distribution of this fish, and I estimated the position of its equatorward or warm edge using a bunch of methods that people in the literature, me included, have used in published papers about species in the move. So the first problem you'll notice is that half of these lines are dashed and half of them are not. Depending on the metric you choose, some of them say, if you fit a linear regression, it's shifted north. Some of them do not. Also, the position of the range edge they estimate varies enormously by like 5 degrees of latitude. So we're looking at a time series here, times on the x-axis, and spaces on the y-axis. So sometimes they put the fish at 36 degrees north, and sometimes they put the fish at 42 degrees north. This is bad. And also, the rates of shift can be different. So they might agree, okay, the species is shifting north, and it's significant, but very much disagree on the rates. So I then asked the question, well, how much data would you really need to get at this accurately? So could more data help us? Could longer time series help us? And the answer, so this is a result from a power analysis simulation, which I'm redoing at the moment, but the answer is for really fast rates of climate change, which is the right-hand panel, you don't need that long of a time series. You need like 15 years to get to a conventional power threshold of 80%, where you're pretty likely to detect the shift accurately if it's happening. But that's more data than almost any studies have that are describing climate change effects. 15 data points is actually a lot. And going to the left on these panels, as the climate effect is slower, as species are shifting slower, you need more and more data. So it's likely that we really are underpowered in our analyses of species in the move. Let me decide how much of what's next I'll cover. Okay, well, I think I'll spare you the Bayesian statistics. I'm so sorry. And I'll end with this stuff, and then I can sort of tell you where my lab is going so that we have time to talk. So going back, so that was a very whirlwind explanation of how I think methods matter for understanding species in the move. So one of the things that I do a lot of now is kind of trying to do the meta science required to look back at our metrics of species in the move and understand how much of the variation we see is just driven by researcher decisions. And if we could take that out, what would the signal actually look like? So that's something I'm super interested in, partly because it gets us closer to a definition of what range edges actually are, which is kind of a weirdly undefined concept. So that's part of the source of variation, I think, in understanding climate change effects on marine ecosystems. But the other source is, I think, some missing theory. So our methods have been a little bit unclear, but so have our hypotheses about what we're actually looking for. So everything I've been telling you about today is reported in things like kilometers per year or degrees north per decade. But as everyone here knows who works on the living marine environment, that's a ridiculous oversimplification of how marine species respond to environmental change, right? The first thing that happens when the environment changes is not for a fish to be like, I'm moving to Oregon. That's just not how marine ecosystems are normal. That's not the pattern of their dynamics. And that's actually not the pattern of any ecosystem's dynamics on Earth. It's just been this really dramatic process, so we've really been looking for it, this global reorganization of biodiversity. But that doesn't mean it's actually a species-only option. And so with a bunch of other kind of species in the move scientists, we've been trying to build clearer frameworks for how to measure these shifts at different scales. So spatially, of course, things could move towards the poles, but you could also move like 5 meters deeper and achieve the same cold temperature you're looking for moving to Oregon. That's not available to all species and in all places, but for many, it's an option. It's a big option on land where we also see a lot of things moving upslope. And mostly on land, but actually this is also an option in the oceans. You can use microhabitats to achieve slightly different thermal environments, which is also a really great adaptive strategy for climate change. And those are just the spatial ones. We have a parallel set of temporal adaptive strategies. Maybe you change your migration timing, maybe you change your seasonality, but species can also change their dial behavior or their foraging behavior to experience different temperatures without moving wholesale to a different part of the oceans. And so what we advocate for in this paper is sort of using more of these spatial temporal gradients when we look for evidence of species in the move. And I'm optimistic that both being clearer about our methods and what we're looking for, and being broader in our thinking of where a species on the move could be going will help us get a much fuller picture of what critters in the ocean are actually doing. These are maps showing you what those gradients look like. So the bottom map is temperature. Gradients of temperature in the ocean are very shallow. What that means is you have to go really far to get someplace that's way colder or hotter. That's not true if you look at elevation. You can achieve a very large temperature change by going a very small distance in elevation or depth. And there are lots more gradients like these that we can map out over space and time and then analyze with kind of new models to look for shifts in a more multidimensional way. So you're lucky or unlucky, I'll skip the Bayesian modeling. But I'll just tell you instead that a big part of what my lab is doing now is trying to do this kind of multi-scale modeling by pulling together data sets at different spatial and temporal scales and modeling how individuals and populations respond to environmental change, and then backing out what that means for species and communities. So I'll skip this part, although I'm happy to answer questions about it, which is a fun spatial Bayesian model of fish that I'm mostly done with. But I'll tell you about a new thing instead, which is highly related, which is a whale project. So because this is, I think, kind of emblematic of the path forward for this field. So what we're trying to do with humpback whales, we're starting this project in earnest next year, is combining at least 3 data sets, maybe more. One of them is tags on humpback whales. These tell us a lot about how whales make fine-scale decisions about when to hang out and eat and when to leave in the context of the environment. That's the right-hand panel. So we also have great historical data on things like krill concentrations around here. These are mostly whales that were tagged in Monterey Bay, some of them further north. And so we can ask, well, how do these animals' fine-scale decisions relate to the environment? But we want to do one better than that and also ask, well, these are social animals. They're part of a population. So rather than just fitting individual-based models, can we also understand how their movements fit into a context of a social network? And to do that, we're using a huge photo database of humpback whales on the West Coast to estimate essentially social affinity between individuals. So understand how their movement relates to the individuals that are present around them. This and almost all of the sort of multi-scale models that I fit are all hierarchical Bayesian models, so that allows us to take really different data sets and use them to inform parameters at different scales. So in this model, there's some stuff that varies by individual whale, there's some stuff that varies over space, some stuff that varies by kind of where you are in a social network. But the point of this is to let us build a model that understands why individual animals make the decisions that they do, but scales up to answer questions like, if there's another heat wave, will the whole humpback whale population in California still come here to forage or not? Which is a really big open question. We don't really know the degree to which humpback whales actually change their foraging in real time in response to environmental conditions versus are operating on memory and kind of longer term cues. So these models allow us to kind of simulate futures at different, from the scale of days and animals making individual decisions to the scale of large populations and seasons, with a lot of opportunities to then inform management. So this is the kind of stuff we do. The thing I skipped is a related model looking at fish distributions over very fine spatial scales in response to short-term and long-term environmental change. So generally, I think that these types of models that merge data at multiple scales and therefore can take advantage of everything we actually know about physiology, bioenergetics, dispersal, behavior at the individual and population scale, but then back out these big picture questions about populations and ecosystems, I think these are the answer to a lot of the questions that I set up in this talk about understanding species of the move. So I'll go back to the outline slide, and then hopefully we'll have some time for questions. So species are on the move, absolutely, and very clearly. but also unpredictably. So we know stuff is moving, but we can't yet tell you what's moving where and why. And when we look for the sort of emergent phenomena we expect to see at the scale of whole fish communities, if species were really closely tracking temperature, we don't see those effects, either at short and long time scales. Some of that's probably because single species are not doing exactly what we think they are in the context of temperature. So marine fish in particular are not as sensitive to temperature at their range edges as we thought. We do think that extreme events are driving a lot of range dynamics and range change, but again, the degree to which we can forecast which species and which extreme events, that's still an open question. Partly because there just aren't that many studies on species range edges. This is like one of my soapboxes. They're really hard to measure because species in the oceans have huge ranges. You never really know you've got the edge. But if we don't understand the edges of species distributions, we don't understand them, period. We don't know why those species are where they are and not elsewhere, or what's making them move. And I think the way forward is by confronting this sort of multi-scale complexity that's inherent in ecosystems and designing better models that can actually capture it. I do this with many people. I gave up on alphabetizing the names and just copy-pasted in the last paper down there in the bottom, right, because they don't all fit. But I am looking forward to your questions. Before I take them, I do want to acknowledge that these are just absolutely colossal and collaborative projects involving whole continents and oceans and many, many wonderful collaborators. So thanks to them and thanks to all of you.
You have to speak into the microphone so that people online can hear your question. Who's got a question?
All right.
Yeah.
Okay, I'm like, I can't hear anything.
But you just woke them up, so well done.
Okay. I'm like, I can't hear nothing, bro. Okay, so first and foremost, thank you for the amazing presentation. It was really cool. So you explained that short-term shifts really don't affect fish movement at all. But you also, I'm not sure if you can elaborate, if you can, for the long-term shift. You basically said that there wasn't enough data to explain it pretty much, right?
Well, we know on average that it's getting warmer. We know on average that fish are moving towards the poles. But if we think it is temperature driven, it would be really nice if from one year to the next when it gets hotter, if the fish move north. And we don't see that. So it's actually kind of a puzzle, right? When we look at data over decades, on average it's getting hotter and on average stuff is moving towards the poles. But in order for us to make a prediction, like for next year or 5 years from now, they would have to be linked in a more kind of immediate and causal way than they seem to be linked in the data. Does that kind of answer your question? Yeah. Awesome.
Yeah.
Maybe like your juvenile white trucks, like they moved up and then they kind of stayed and not continued into moving.
Yeah.
Exactly.
Shorten up and then moving back.
And then maybe
Yeah.
there'll be another big jump something like that, right? So.
Okay.
That's a great example.
Thank you so much for that presentation. It was really compelling and so interesting and thoughtful. And yeah, I think a lot of the things that you bring up are applicable to folks in this room that study all variety of parts of the ocean. My question is in regards to temperature specifically and the scale at which we're able to measure oceanographic conditions.
Yeah.
And whether you have expectations about how to reconcile like local and regional microenvironments within the ocean, and especially in large data sets, rectifying that we can only really extrapolate things from sea surface temperature, and these fish are experiencing the whole water column, and how that plays into your work moving forward. Because a lot of these things are, I think about these things a lot as well, and the sea surface temperature issue is a pain in the butt.
So this is such a good question. I think it's the #1 conversation I have with particular grad students, because I talk to you a lot about like which species metric do I use, but the question is always which temperature do I use? We all think temperature is driving so much about ecology, but which temperature? Is it daily? Is it hourly? Is it monthly? Is it maximum or minimum? Where did it get measured from? Is it from a model? Is it from an instrument? And this is, again, not something that we have great rules of thumb for. In bottom trawl surveys, so there are a couple advantages to using bottom fish data, which is another reason why I've used it so much. In addition to the fact it's just a huge data set. Some of them actually carry thermometers or do in situ temperature measurement, and then we can actually analyze the temperature where they found the fish. And also, because they're on the bottom, there are ocean models that have come online really recently that have pretty good reconstruction of bottom temperature on continental shelves. The one that most people use is called GLORIES. It's a European data product. So that's what we've used for a lot of like the marine heat wave, demersal fish stuff. But if you work on something pelagic or highly migratory, where in the water column are you looking and when is a really big question. I think that ideally, I think it's helpful to start with what you think is the temperature the species is really responding to, and then get as close as you can on the data side. But that's a non-trivial step to try to find the right data to match, especially at these big scales. I don't use just like overall average sea surface temperature very much, but also in the oceans, these temperatures are pretty correlated, so it's not the end of the world if that's what you use. Does that kind of help?
Yeah,
okay.
Definitely. Thank you.
I may have missed this, but one of the things, a lot of these animals don't necessarily respond immediately. Did you try different time lags in the data based on their life history, or?
So this is a great question. I've looked at this in different ways in different projects. In the marine heat wave paper, we did look at time lags at the scale of the whole community. So like, does the biomass of the community or the assemblage lag the marine heat wave by 1 or 2 or 3 or however many years? And the answer to that is no. It's likely, though, that there are species-specific time lags, so that a species with a short life history might have a lag of a couple years, a species with kind of a longer life history might have a lag of many more years. That's partly what the empirical dynamic modeling project my brilliant postdoc, Emma Walker, has been doing, was meant to detect, and it hasn't really seen that. But part of the problem is that for things that have a long life history, in those kinds of more sophisticated analytical tools, you need to have the events just happen a bunch of times for the model to say, okay, I'm really sure there is a lag effect. And if if something's lifespan is 30 years, you don't have recurrent generations experiencing these things enough. So I think it is a little bit of a data limited problem, but it doesn't kind of magically fix the models to put it in. It's a great question.
I'll ask one. Are you seeing, and then, oops, sorry, then I'll come back this way. Are you seeing the fish moving deeper at all, like from those bottom trawl surveys? Are you seeing that in addition to the temperature shift? Is it more or less predictable?
Some of them are not. And interestingly, so one of the places where fish deepening has been happening really prominently is the North Sea. So if you go look at that survey or those surveys, they are going deeper. It's often a combination of like a rebuilding from overfishing. So often when you see fish assemblages ground fish going a lot deeper, what happened is like they got really hammered and then they rebuild but deeper and colder. We don't see overall that everything is going deeper and down the shelf. And the shelves all look different. Our shelf is really steep. On the East Coast, it's much, so their opportunities are really different by survey, which is probably why they're not, but it's not as universal of a pattern. The deepening is happening, but it's not quite as universal as shifting poleward.
Great talk.
Thank you.
So my question is a little more similar to Legita's question earlier. So I'm curious that when you look at the short-term and long-term changes in your range of models, have you included the turnover time span of different species? So like that includes their lifespan and life cycle. I know you said you didn't, but because with that, we might be able to see how well they might move versus how well they might adapt or evolve.
Yeah.
For example, like species with shorter life history, are they changing genetically as a whole to become more tolerant?
Yeah.
And when you standardize it based on this scale at which they live, then do you see more significant changes?
So this is such a great question. I think it kind of intersects with every part of the stuff that we talked about. So when we talk about available gradients that you can measure and model, the gradients obviously depend on a species' life history, right? Some of those distances are just too big for a frog is not going to march out of the tropics. That's just not going to happen. So for a frog, you're really focused on microhabitats. So yes, obviously we want to tailor these analyses to what's in the capacity of a species' life history. And I actually think the clearest place to disentangle that stuff is marine invertebrates, because marine invertebrates range extensions are often driven by ocean currents, not as much by their own movement, although that's not always true. And so, for example, the species that I said 100% of the range extensions in and out of California are during El Niños, those are species that have really, really long pelagic life stages that just kind of hang out and get carried around by the current. So it makes sense that during an El Niño they just pop up, versus other ones where they don't have that life history, that doesn't happen. And actually there's some interesting examples that for things with shorter larval durations, they do have a consistent but lagged El Niño response, that you find the adults like 5 years after the El Niño, because they dispersed a little bit up the coast, but then we didn't see them until they grew up. So this is a big part of the puzzle. It's just hard to know how to kind of scale it from thinking one species at a time to these very large multi-species analyses. Yeah.
Analyze them separately, like by similar lifespan and life history kind of case, and would you see more effects based on that, or have you done that and have you seen more effects based on that?
So this is a great question. We've tried it for fish, and the challenge is always getting those data and getting them in the form that we have the observational data. So for fish, the trait data and databases are better. So the fish stuff that I presented uses up to like 1,500 or 2,000 taxa, so it's a lot of species, but we can merge those with trait databases and ask questions like, do we see differences by feeding guild or by trophic level, or by whether they're primarily pelagic or demersal or something. And generally that doesn't help. There's sort of a separate interesting literature that often when you do these big analyses and then you like chuck a bunch of traits into the models, it never helps. And that's very discouraging. We would love it if it helped. So people have been trying to understand why don't traits generally help. And I think the answer is you do need to sort of think, well, for this species, here's what makes sense. And for marine invertebrates, the trait databases are not as far along, so we can't do that. We can use taxonomy as a proxy for traits, so that's something that we do. So I have a project looking at East Coast invertebrate range shifts where that I didn't tell you about, where we're sort of using the order and the family as a proxy for their life history, because we don't have a spreadsheet that says for each of these 150 species what their life history is. But merging those things is quite challenging and an active research area. It's a great question.
Any final questions for our speaker? All right, if not, you're welcome to come up and talk to her individually. And let's thank Alexa for that wonderful talk.
Thank you.